Biology Part 145. Fibrocartilage has a matrix containing prominent bundles of collagen fibers and occurs in vertebral discs. It combines strength and rigidity. As its name suggests, elastic cartilage contains a high proportion of elastic fibers. It contains a high proportion of elastic fibers. It is found in external pinnae of the ears and in the epiglottis. Bone. The matrix produced with a bone-forming cell or osteoblast. It is called osteoid. It contains collagen and osteoconnection proteins, which are thought to act as sites for the growth of inorganic crystals. At first, the osteoid matrix, uncalcified and insoluble minerals are rapidly deposited from the blood. The commonest bone minerals are the hydroxyapatites formed from the calcium and the phosphate ions. These have the general formula Ca10PO4 6OH2 and form an extremely hot crystalline matrix. Other ions including carbonate, magnesium and fluoride may also contribute to bone structure. Once trapped in the hot matrix, the bone cells, now called osteocytes, are unable to divide. Each occupies a small place or lacuna, as in the case of cartilage. But unlike chondrocytes, the osteocytes are linked to the neighboring cells by cytoplasmic threads, which lie inside tiny channels known as canaliculi. It might be thought that the bone tissue would become permanent once formed. Nothing could be further from the truth. A second type of the bone cells, called osteoblast, continually erode the bone matrix. The osteoclasts are large multinucleate cells similar to those of the macrophage. Like other macrophages, they develop from the monocytes. Working in small groups, osteocytes tunnel through the bone, leaving the cavities which are invaded by blood capillaries and are new bone forming the osteoblast. The process is illustrated in the figure. Within the tunnel, a new bone matrix is laid down, the concentric rings forming an arrangement known as Haussian system. The results of the activity are clearly seen in the section of compact bone. Note that new tunnels have completed the Haussian system, while older system may have been cut through and partly destroyed. The factors which regulate the balance between the bone destruction and rebuilding are not fully understood, but it does appear that the osteoblast and the osteoclast are able to respond to the mechanical stresses, so that the shape of the bones are determined by the load placed upon them. Constant use of the leads and the strengthening of the bones, while disuse results in the weakness. In this way, the bones of the skeletons are constantly remodeled. In an adult mammal, 5 to 10% of the bone is replaced each year. It does not seem surprising that every person has a skeleton of the correct size, of the proportions to provide support, but you should appreciate that this can only be achieved by means of the sophisticated internal control mechanism. When conditions are altered dramatically, the existence of such mechanism becomes more obvious. For example, exposed to weightless conditions, astronauts start to lose large amounts of calcium. Human skeleton development. The bones of the human skeleton originate in two ways: the thin bony plates of the skull and the and parts of some other bones are formed directly by the clusters of osteoblasts which appear inside fibrous membranes. The strands of the bone formed by different clusters are called tuberculi, single tuberculi, and become linked to the form of a loose network described spongy bone. The whole process is known as the intramembranous ossification. As development continues, the remodeling of skull plates converts into some of the spongy bone, compact bone, and allows the plate to reach their adult size and shape. The second method of bone formulation involves the replacement of cartilage and is called endochondral ossification. As stated earlier, the skeleton of the vertebrate embryo consists mainly of the hyaline cartilage. Each cartilage element is surrounded by the connective tissue jacket. The perichondrium is the same shape as the bone, which will replace, as shown in the figure. The cartilage acts as a model for bone formation. Ossification of bones like long bones of the arms and legs begin with the blocked vessels, penetrance, and the purchondrium midway along the shaft or the diaphysis of the cartilage as shown in the figure. This stimulates some of the cells of the perichondrium become osteoblasts, which produce a collar of compact bone in the shaft region. The jacket of the connective tissue covering the development bone is called the periosteum. A primary ossification center appears inside the shaft and progressively invaded by the blood vessels and osteoclast. The matrix of the cartilage tends to become calcified in the deposition of calcium and phosphate, but it is eroded by the osteoclast, leaving spaces which eventually fuse to form the narrow cavity. Cartilage continues to grow and either in producing an increase in length, while most of it is later replaced by the spongy bone. I'll tell you more about the same in my next video. That's all for right now.